Yes, I think we're going a bit into the issue, a bit of the meeting today, so I'll start quickly. Do I do it this way? Yeah, here it is. So I can't see very much, but you can see it, and it'll be okay. This is just an image of the B cell receptor signaling that is a bit the topic of the meeting today, and the role of antigen stimulation, which is a key point, and what it triggers below the surface of the cell and different tyrosine kinase that can be phosphorylated according to antigen stimulation. And we start seeing here the role of CD19, which maybe we'll not talk about that much, but can obviously be a target of some forms of treatment. You see BTK here, PI3 kinase here too. So this is a bit the model where we will fit the discussion in this meeting. And if we go a bit more in detail, you see the B cell receptor is composed of a membrane IgM bonded to the CD79AB heterodimer. And when there's a IgM ligation by antigen, this induces tyrosine phosphorylation. And this recruits a number of kinases that we just saw. So this is a scenario that we're actually working in. And if I go back a few years, just to try to tie different aspects, we learned many years ago that CLL, despite being a single disease, has two very important features that was in fact shown by gene expression profiling, that you can have unmutated and mutated forms of CLL. Now this opened a big debate on whether it was one disease, two diseases years ago, but we learned it was one disease, and this was proven in fact by gene expression profiling. You see CLL is clearly one disease, but if you pose a different question to see if they're differently mutated and unmutated, you saw that they're the different gene expression profiles. So we have two subgroups within a single disease. Why this was important was clinically. If we tie it to patient, that patient with an unmutated immunoglobulin profile do worse than patient with a mutated profile. And this is still today one of the most robust prognostic markers, biological prognostic markers in CLL. So this is relevant, and I put it, although it's old data, in respect of the fact that with new drugs, this is changing. And I think it's very important. This obviously with conventional treatment, chemoimmunotherapy. And there are biological data to support this that go back a few years. I mean, here you see the unmutated versus mutated. And if you stimulate these cells to the B cell receptor with IgM stimulation, you can see the response is evident in the unmutated, while it's not in the mutated patient. So they respond much less to antigen. And that is one of the reasons for actually doing better clinically. And it has been shown in different ways. And the bottom line is here, mutated, here, antigen stimulation. There's a big difference in signaling if you're unmutated compared to mutated. So this is a main point why the unmutated have a more active disease, proliferate more, and probably respond less well to conventional treatment. Even if you do gene expression signaling, there's much more modulation of genes in the unmutated than in the mutated cases. I think I have another slide here. This puts together, in fact, not only the mutated and unmutated, but also patients clinically with progressive disease versus stable disease. And again, if you trigger them with IgM or with IgD, indeed, the unmutated, you see there's a clear difference, not in the mutated. And even if you look clinically, progressive disease, stable disease. So there's a tie between the biology of the disease, response to antigens, and disease aggressiveness. So that, I think, is a background, and clearly we have drugs now that target this, and this is, I think, one of the topics of the meeting today. We have ibrutinib up here, we have idelalisib here, and more are coming, and more will come, that are actually targeting these tyrosine kinases that have a key role following antigenic stimulation. There's another piece of information just showing the response of primary CLL cells to in vitro treatment with ibrutinib. Here's comparing CLL to healthy donor B cells, and you can see that the stimulation is greater in CLL cells than normal B cells. And if you look at unmutated versus mutated, and the unmutated is the yellow, you see again the unmutated in fact respond even better in vitro than the mutated to ibrutinib stimulation response. So you see IgV unmutated CLL shows significantly higher susceptibility to ibrutinib than the mutated cells. So this is a background scenario, and what is happening with the drugs. So we have emerging treatment strategies for these diseases, targeting the B cell receptor pathway. BTK inhibitors, ibrutinib has clearly opened the way. I don't have to underline this much more, but the new drugs coming, obviously. We have PI3 kinase delta inhibitor, idelalisib began leading the way. Let's not forget that we can have other mechanism-based drugs, and the BCL2 pathway is obviously a key target in CLL in particular, but not only, I mean, lymphomas too. And the second generation anti-BCL2 ABT-199 is certainly very interesting and promising, and I think it will come up probably in talks today and tomorrow, with data recently published. And then your antibodies, apart from the usual antibody, let's remember, for instance, blinatumomab. I mean, this is a bispecific monoclonal antibody targeting CD19 as a target, but then the mechanism is mediated by CD3, so it is a form of immunotherapy. This developed more in ALL with very interesting data, but it is started lymphoma, in fact. This is a technology, and this will certainly find space in chronic B-cell malignancies. So I think this is a background where we are, and I wanted just to put this in a clinical scenario. What is indication, at least in Europe? I mean, here in Latin America, realities are different across the world. So this is what the situation is in Europe. So last year, now, Europe is not United States of United Nations, United States of America. We are not United States of Europe. We are 28 countries. So EMA, which is what we used to be EMEA, EMA is in London. And once EMA approves, it's not like FDA. We are not states. So then after that, every member state has to review and reapprove, which is sometimes a very cumbersome process. This is because of, I said, every country has its own rules, which makes it a problem. I just give you one practical example. We just got approval. No, let's go back this way. Let's see what had happened. This is in 2014. So we're talking about a year and a half ago, because I think it was September 2014, if I remember well, when both idelalisib and ibrutinib got approval in Europe. So that is, now we are in February of 2016. So in Italy, we just got ibrutinib approval now, just to give an example. So this is because of negotiations. Largely, let's be very open, because I think this is the purpose of the meeting, largely discussing costs of drugs. This is a key point that we're all facing. So this indication in Europe, and I'm saying this because I'll come back to this. I mean, we can only give drugs on indication. So there's no off-label for us anymore, maybe years ago. Now this doesn't exist. So indication in Europe is the same for idelalisib and ibrutinib. Idelalisib is not alone. It was rituximab. As you know, ibrutinib is alone. Indication of relapsed CLL plus rituximab for idelalisib. And this is the extended indication, CLL patients with a mutated p53 or deletion 17 as first line. So I'm saying this as extended indication because when both Janssen and Gilead went to EMA, they went for second line approval. And this was surprisingly an indication that came from the regulatory agency, that based on the poor data in the 17p patients and on encouraging data that were already present there, EMA decided to extend the indication for both drugs, which was obviously very important for patients. So that was very good. So regulatory agents can work in favor, not always against. I'm saying that this doesn't happen very frequently. So that is what we have in Europe. Now the timing of realization has been very different in different member state countries for what I just said. And then there are others, but we won't go into this. So this is for CLL. And then we have non-Hodgkin lymphoma, and you see here, I don't know why they're two different colors, but anyhow, idelalisib for patients with follicular lymphoma whose disease has not responded to 2 previous lines of treatment, and ibrutinib for adult patients with mantle cell lymphoma whose disease does not respond or has come back after previous treatment. So these are the indications that we have in Europe. Then there are obviously many issues. I just put the slide from the recent international workshop on CLL. It was held in Sydney in September. And there was a debate in a symposium, sorry. There were 2 events, in fact, in the main program. One was a debate saying, will novel target agents replace chemotherapy in the manner of CLL? And obviously, being a debate, there was one speaker in favor of saying yes, the other one no. So this just to underline how it is felt that the new drugs could in fact change the whole scenario of management of CLL, which I think is one of the topics of discussion in this meeting right here. The other interesting is symposium that was in fact sponsored by Janssen in the main program. And Janssen itself put this topic and said keeping pace with treatment advance associated costs. So it's not only us that are worried or our administrators, it's also companies are obviously concerned on this issue. Because there is this growing issue of sustainability and accessibility to drugs and technologies. I mean, if we have first line treatment for 17p and p53, which is my next slide, are we testing patients? So I'll go to the next slide. So I put a few points for your consideration, and which I think will be some of the topics that we will discuss in this meeting. One is clearly the first, which goes back to a debate in Sydney. Will chemotherapy be replaced by the new mechanism-based drugs? I think this is one of the key topics. In the meeting, data will come out from Resonate 2 showing first-line treatment, and certainly there will be approved ibrutinib for first-line. So the question is, will it replace? The second question, it's all for the discussion. Could chemotherapy be spared? And if that's the case, for which patients? For all or for some patients? In this same scenario, what is or will be the role of anti-CD20 monoclonal antibodies? Because you could have sparing of chemoimmunotherapy, you could have sparing only of chemotherapy. Maybe the anti-CD20 could still be part of the backbone of treatment, question mark. These were two question marks. Can some patients be spared the new drugs? Let's turn it around. I mean, one is, can we replace the conventional treatment with new drugs, which is obviously fashionable? Yes. If we turn it around, could some patient be spared the new drugs? And in fact, we included this in the program later on today or tomorrow, I can't remember. Then this is a key point I just mentioned. Since we have this indication in Europe where for first-line treatment, 17p and p53 patient can be given the new drugs, then it's a key point that we tested the patients. And this is not happening in all patients, obviously. We know that, and this will again be discussed, that you can have patients that do not have a 17p deletion but have a p53 mutation. So I think in real life, at the time of treatment, we should at least do FISH first. But in theory, if FISH is negative, we should sequence. Now, who is doing that? Even in Italy, not all patients have it done. So let's be very practical. This is for first-line treatment. And what about relapse? We know that the incidence of let's say of p53 disruption, relapse is much higher, 35, 40%. So in an ideal world, every time we treat a patient, we should be doing these tests. Is this doable? This I put it for the panel. And I added here another important point, at all ages. In the old days, what we do with a 75-year-old, is 75 old? Today it's changing. I mean, I come from a country where the median life expectancy is well over 80 for women, and even for men it's over 80, luckily. So things are changing, and the age is advancing. So a number of CLL patients getting bigger because of an age issue. So in the old days, what we do in an 80-year-old, we wouldn't test anything for biology. What would we do? Maybe with a chlorambucil. Now it's different. Now we should test. And who's going to do 17p or p3 in an 85-year-old patient? This already said, the major issue of accessibility and sustainability. And then I think, again, for discussion on label and off-label use. I mean, I was in India last month. We had an AHA event there, and there was a symposium. In fact, they asked me to talk about CLL, and ibrutinib is in India, but there's no indication. So it's just a matter of who can actually get it. I'll put it that way. So I just put it for the question, and they asked me, what do you do in Europe? And I said, well, we go by indication. We can't give a drug out of indication. If EMA and our regulator says ABC, that is what we can do. Irrespective of who pays or pays, all patients get the same. And that's it. And that is it. Thank you very much. Carlos.
Thank you, Robin, for your presentation. But I think you allude a very important point. Besides the scientific environment, you talk about the regulatory agencies in the US and EMA. You know, in our countries, I can say particularly in Brazil, we have a lot of problems with this kind of agents. And the financial for these new drugs is the main cause. Uh, Tell us, how is that, how it works? If you have the approval of EMA, it's not necessary the different countries accept this approval. They rediscuss the indications based probably not in a scientific evidence, but in a local, in a the possibility of having funds for that. Can you allude a little bit on that? And how is your point of view in the future? How should we deal with these agents? My opinion is for sure you have to niche as much as possible the indication of these new drugs. Can you talk on that?
I will try, Carlos. No, thank you. I think this is a very important point because we in Europe have gone through this problem. I mean, as I mentioned, since we're talking about these new drugs here, these were approved, both the idelalisib and ibrutinib, in September of 2014. And in Italy, as I said, ibrutinib is available only in the last month or so. This is because, I mean, I can't speak for all 28 countries in Europe. There are differences, obviously. For Italy, the issue is that you go through, we have a regulatory agency in Italy which is called AIFA. They will not go into the indications. The indications, you know, that they have to be the same in Europe. The problem is obviously whether to approve or not. It's not that you get approval in London, it's officially approved everywhere in Europe. It has to be approved in every local country. In Italy, it's because we have a national health system, so any patient, whether he is black, white, red, yellow, rich or not rich, will get the same treatment so far. And until a country doesn't go bankrupt, this is a reality, fortunately, and this is very positive, obviously. So once it's approved, it's the same for everybody. So the indication will change. I think it's a lot to do with the importance of the drugs and the costs. So there are a lot of negotiation on actually the cost of the drug. So that is the key point. To give you a practical example, ofatumumab, J101 has not been approved yet in Italy. And I can imagine, listen, I'm just speaking loud. I don't know officially, but I'm saying, why isn't it approved? I can imagine that they haven't approved because they already have 2 anti-CD20s. So I imagine that the agency said, listen, we have already 2 anti-CD20s. Do we need a third anti-CD20, which we can discuss? I mean, that's a different point. I'll bring it up in one talk, but that's different. Other drugs are much more important. So these drugs are much more important. We didn't have any B-cell receptor antagonists available. So these go through quickly. So discussion has been on the cost of the drug. It's very different. So two different. One is the necessity of a drug, and the other is the cost. Other countries in Europe are different. I know of a couple of countries, I think the UK and France, where the government has set aside a lump of money. So if an important drug comes, then you can use it immediately. But it has to be a very innovative drug. Not all drugs approved are the same level of, let's say, innovation. Some are really revolutionary, some less. So it has to be probably balanced, which is not easy. A final point I may say is that we luckily for many months in Italy had what we call a name patient program. So prior to approval, we managed to treat in Italy, I don't remember exactly figures, but I think over 500 patients with CLL with ibrutinib. Luckily, and Janssen did this program, and Mark can say much more about it, in many countries in Europe. And this has been extremely important because in waiting to have the drug officially, we could treat many hundreds of patients with ibrutinib in real life, which again is very valuable. Now we're doing a database and putting data together to see how hematology using ibrutinib have used, in fact, ibrutinib in real life. So that has helped in the bridge in the window between having EMA approval and local approval.
So I also think that most of these newer drugs, especially for CLL, they're given chronically, they're given orally, and they're given until the disease grows. And I think that these kinds of trials are ongoing now, but it may be that if we cycle these with chemotherapy for more limited periods of time, that we may be able to, get uh, 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 a lot of activity out of them and not give them chronically, certainly not give them chronically to every single person. And I think that these are the kinds of trials that are also critical because they're going to have huge impacts on obviously cost.
Yeah, that's it. Again, I think it'll come up clearly in the talk, but that's a very important point. I mean, can we reduce the length of treatment? I mean, I'll bring this up the other way around in the talk sometimes. And can some patient be spared, or can they maybe be quote unquote cured with conventional treatment? Which, if yes, which patient? The other is obviously the new drugs in combination with something else. Can we then stop treatment? Can they become MRD negative? And this will be, I think, it'll come out.
And one other just question for the group here, because I thought it was interesting, there may be, again, subsets as we talk about who may really do poorly with what we have now that these new therapies can, the P53 being in the unmutated group, are two examples. So how much if those become, like a subpopulation that these drugs are very effective in, how much access do the countries have to do those kind of testing for it? Again, as you mentioned, you're bringing for potentially looking at the ABC. There's a new nanostring technology, but what about Dr. Kaminsky within Argentina? If you look at the different hospitals on getting P53 or doing sequence of P53 versus the FISH, what's the access for those kind of testing? If that is, again, the right population just should be getting these therapies early.
I think
How about some of the other countries? Mexico, I mean, what kind of access do your patients have potentially for those testings?
almost nobody has a proper genetic assessment. The fish is not available in the country, and unless you treat patients outside the public system, uh, and people who have private insurances, then we do those tests. But in most of the institutions, as I will show you later on, they don't do any assessment at all.
So
Yeah, no, this is, I mean, it's, that is the key point, because, I mean, I don't know, in other countries, is the indication first line with 17p only in Europe? Is it in many countries?
it's in the United States.
U.S. actually, Robin,
Yeah.
FDA did the same thing that
They
EMA
the same,
did.
so it was
Yeah, but it did only for 17p, not for p53.
Yeah, right. They did it
Okay,
for
so that's a
17.
major,
So we have some,
17.
major point of discussion.
Oh, really? You mean in the EMA, it was for mutations too?
Also
Yeah,
the mutation.
Oh,
yeah.
okay, yeah,
It's both.
yeah,
There was
yeah,
a-
both.
both,
All
yeah.
right. Yeah.
And for idelalisib, it did not get a frontline 17p indication in the United States.
only
Only for
brutal.
Did not.
ibrutinib. It's
Only ibrutinib.
Yeah, but from a practical standpoint and getting it reimbursed, insurers don't make a distinction between del17p and whether there's p53 sequencing. They don't even know what that means. So it's, it's covered.
And I can to give you a practical example how countries around the world work. Turkey, which is not a small country, a few years they couldn't even give rituximab. FCR was not given until recently. Then the government approved it, but they said you can give FCR if the patient just think of the reason behind it. If the patient does not have a 17p deletion, this is a fantastic. Now, why did they do that? Because how many patients in Turkey will do fish on a CLL? This is a government decision in Turkey.
Well, I also think that we need, well, you already alluded to this, Robin. Not all upfront 17P have the same outcome. Some of them actually can do relatively well, but we know that if you take the entire group and you do the randomized study, the people who are not getting ibrutinib do not live as long. So I guess we just need to have a better understanding of who truly upfront needs it.
Here we're talking about patients who need treatment. I mean, I wouldn't do it. Yeah, that's key.
So Robin, can you comment then, because in many places you cannot do 17p testing, right? But can you comment on how you maybe can identify empirically who might be, more likely to have 17p? For example, those patients who had FCR frontline or BR or FC, those patients who fail within a year, progress within a year, it's likely that's an enriched patient population. Can you comment on that? Because it's an empiric way of trying to pick out poor prognosis patients.
In theory, for relapsed patients, you don't need the genetics because we have the indication, so broad indication. So that would not be necessary. If you test in early relapsed, you can have over 40% of patients having a 17p or p53 mutation. So I think, empirically, you could happily give the new drugs. And I think the question is that in a patient, we've done this in other meetings, how would you treat a patient with let's say, an early relapse? Now, early, is that 3 years or whatever? We can discuss that. But anyhow, let's say within 3 years, and everybody would say we go to new drugs, if available, obviously, irrespective of the genetics. Clearly, it becomes fundamentally in first-line treatment. If you have patient progress, need treatment, and we can give the new drugs only if you have a 17p or a p53. So that, I think, is an unmet need. We're trying in Europe to do this, obviously, as much as possible, but the world is very broad. So, and as we heard, this is obviously very different in different parts of the world.
I think that, for patients who have a short remission duration after a good chemoimmunotherapy regimen, as you mentioned, yeah, sure, they probably would be overrepresented by 17p, but in a lot of ways it's irrelevant because biologically the patient has already told you that they're going to do poorly. And remember, in all of our trials with these agents in the relapse setting, they weren't just patients who had relapse disease. They were patients who had bad characteristics, either progressing within 2 or 3 years of having, say, FCR. So these trials do not include the typical patient, somebody who gets FCR and has a 4 to 6 year response duration. And I think we kind of forget that. The other thing I'd mention is we always talk about this 40% figure of del17p in relapse setting. That data is 40% of patients who are fludarabine refractory, right? Not fludarabine exposed. Fludarabine refractory patients have del17p. If you surveyed the whole population of previously treated patients, it's certainly not going to be that high. And I think I would guess that in the future, we're going to change the natural history of this disease by treating with more effective drugs up front. And we're not going to see as much 17p deletion. It's kind of like, how much blast crisis CML do you see these days? Vanishingly rare, fortunately, because of drugs like Gleevec. So I think all these things we have to keep in our mind as we move forward and we face these challenges of limited access to these expensive drugs, and how do we then triage who we really try to treat with them.
Stephen, can I ask you very quickly? We're out of time, but in the US, all 85-year-old up are tested for 17p?
No. I mean, it's, most patients are treated by community oncologists, not even hematologists, oncologists. And so what they do is all over the map.
Okay.
Yeah.